fungi that even helped plants make it onto land millions of years ago. And that the mycorrhizal fungi, most plants have that association and are definitely benefiting from it. So a lot of what most people don't really think too much about fungi because most of the time they're just all under the soil and only rarely do they pop up, so to speak, with the fruiting body. But under the ground in the soil, they are performing vital functions for plants and forest ecosystems, agriculture systems, very, very important. So here is a schematic or figure trying to show a couple of concepts here that if you think about agricultural systems, very often what they do with conventional farming is they apply synthetic fertilizers, inorganic fertilizers, minerals. And what they're trying to show is that, yes, that those minerals are in the form that are ready to go and can immediately be taken up by the plant. And then kind of in contrast to that is organic fertilizers that are complex materials that are applied into the soil, and they need to be broken down first before the plant can get access to them. And so the microorganisms in the soil are important for degrading that organic matter, releasing and recycling those minerals so that the plant can then take them up. And so the mycorrhizal fungi are playing a pivotal role in that, as well as many other microorganisms, bacteria included, that you need to have a good mixture of microorganisms in the soil system in order for plants to thrive. And it's a more steady state, long-term process when you have organic fertilizer. If you overdo it with synthetic fertilizers, one of the risks you actually can encounter is toxicities in the soil in terms of too much salt production or accumulation, I should say. And you can get that causing problems with the plants as well as the microbes. And so I wanted to talk, as long as we're talking about mycorrhizal fungi and how, they are growing out off the roots into the soil system, and they're trying to access all those lovely minerals and the water, especially into areas that the roots just can't physically get to. I wanted to talk a little bit about the soil, since we haven't had a chance to do that in this class yet. And so certainly, I mean, we offer a whole soils class that you're all botany majors are all asked to take because it's that important. I mean, you have to know the soil system because that's what the typical plant lives half its life, half its body, I should say, half its body is in, more than half its body typically is in the soil. So knowing a little bit about that environment. So I am not going to replicate, obviously, in one little snippet of a lecture what you learn in soils, but there's a few key things that I want you to grasp. And so when you think about the solid matrix of the soil, you certainly have inorganic particles that serve as a reservoir of these minerals, these nutrients. So these mineral nutrients like potassium and calcium, magnesium, they are a reservoir. It's like a bank of these minerals that the plant will, extract from. And then there's certainly a mixture of organic particles in the soil system as well that provide the nitrogen in particular, and the phosphorus and the sulfur. So the microbes are busy recycling that organic material, releasing those minerals, and then the plant is able to get access to them. So there's all kinds of different soil systems. It's in mixtures, ranging from the very coarse particle size of sand all the way down to the very fine particle size of clay. And certainly roots have a very hard time if the soil is too much clay, roots have a very hard time growing through that. It's too compacted. On the other hand, if it's too much sand, then the water and minerals tend to leach through, and the plant can't get quick enough access to all of that to stay healthy. And so you definitely want a mixture. And I'm not really going to get into the soil types. Basically, in this section, I want to get you thinking about how are the microorganisms in the plant getting access to the key minerals and the water. And so here, certainly when you look at a soil system, if you think about a rain that has occurred, you definitely have water in the soil system. You have dissolved gases in the water. So the same gases that you have up above ground, you have below ground. You still have all those mixtures of gases down there too. And hopefully the soil is loose enough where you have some nice loft to it so the roots can easily grow through it. And then you'll have pockets of air, which is necessary. You don't always want the soil saturated. That's not good for the roots because it's hard for them to get access to oxygen if they're always saturated with water, the soil around them. So that's really where the plant is mining for minerals. It's in the soil system. Their roots are needed for that. And when you examine the different soil systems, basically the soil particles have a net negative charge. So I need you to know that. All soil particles tend to have a net negative charge. And so if you remember from basic chemistry, that means that positively charged minerals can stick to that, so to speak. They're attracted to the negative charges. And so they have cations, positively charged ions, or minerals that can bond with the soil particles. So that's your reservoir. So all the cations, all those types of minerals that are positively charged, there is a reservoir for the plant. As its roots are growing through that, they'll have access to that eventually. And so C+ is just abbreviation for cations. So it's very, very difficult for the root to directly get access to cations that are bonded with the soil particles. That's challenging. And so really the roots don't do that. They will get access to the minerals when the minerals are dissolved in the soil water. Then the plant has an easy time of taking up, relatively easy time of taking up those minerals. And so basically, when you have all these cations stuck, bonded to the negatively charged soil particles, the roots, the plant has to kind of wait until those minerals get released from the soil particles, get dissolved in the water in the soil, and then the plant can take them up. So that's what this image is trying to show. This left image is the soil particles with all the different green balls or different minerals, positively charged minerals, and then they're trying to show that they get released off into the water, and then the plant root can take it up. So if the plant root has a hard time getting direct access to those bound particles, what is going on here? And so if you think about, how the minerals can get released, break that positive negative bond, there is a process called cation exchange, which if you've taken the soils class, I'm sure you've learned about that, probably in more detail than what I'm going to say here. But basically, this is the idea that you have the minerals coming off of the soil particles in a particular way based on their affinity, how tightly they bind to the soil particles. So some are more weakly bound to the soil particles, some are strongly bound. And if you can imagine rainwater percolating through the soil, you'll get this cation exchange property where you can get, when they say exchange, what they mean is one mineral can bump off another mineral off the soil particle and take its place. So it's an exchange. And so we have, let's see if I can find it in here. This one. You can look up these lyotropic series, they're called. And so this is an example of one where the way you interpret this series is working your way from the far left is really high binding affinity to the soil particles. And working your way off down the line here, off to the right, you get weaker and weaker binding affinity. So with the cation exchange, what this means is, let's say magnesium is bound to the soil particle. And then as it's raining and water's moving through the soil, maybe there's some dissolved hydrogen atoms. And so that hydrogen can bump off magnesium because it's higher in the Lyotrophic series. It has a tighter binding. So if hydrogen comes along that soil particle, it can bump off magnesium and take its place. Whereas it can't really, like, the reverse can't really happen because magnesium is lower in the series here. Does that make sense? Okay. So aluminum, unfortunately, really, well, it's okay that aluminum is tightly bound, actually. You don't really want aluminum loose in the soil system, the water, if you can, not too much of it anyway, that's really not good. Aluminum's toxic, to plants, animals. So this figure that they're showing, they're trying to show a clay particle with all the negative surface charges and all the mineral cations bound to it. One thing that's important to note is that protons, H plus, that cation is fairly abundant in the soil system for several reasons. Here, they're trying to show that as the root is going through respiration, just like us, they're going to give off CO2 when they do that. And when you mix CO2 and water together, you end up with this reaction, which gives you free protons, H plus. And so there's all kinds of microorganisms in the soil doing similar things where they're exhaling, so to speak, CO2. And so you can get a nice buildup of protons in the soil system. And so that's good because that helps knock off, so to speak, some of these other cations into the soil water.
Like natural form of like antioxidants for like plants, or is that not how they work also?
As far as this,
Yeah, for like process, giving like charged particles more electrons so that they're neutral.
I mean, you can mess with this idea in the soil system. So like if you get your soil tested and they say, you know, there's too much of this or that, there's ways to amend your soil with certain mixtures of chemicals. You can do that. It depends on what the problem is, so to speak. So a lot of it has to do with, in practical terms, a lot of it has to do with managing the pH of the system. And also just one of the mineral nutrients that plants need a good amount of is phosphorus. But phosphorus is negatively charged, which I need to talk about that because then it can't stick to a clay or, I mean, to a soil particle because a negative and a negative won't interact. So with this Lyotrophic series, you can see that certain cations can get pretty easily bumped off the soil and get replaced, exchanged for another. And so you have to just, you know, imagine this is a very dynamic system with water movement through the soil. You can get this cation exchange process going on. And like I said, this is not a soils class, so I'm not going to get into huge detail on that. So here again, here's your cation exchange idea on the upper right figure here. And then the other idea of how do they get access to these minerals is microbes, including mycorrhizal fungi. And so the microbes are capable of releasing certain chemicals, certain enzymes that can help get access to these key minerals so that the plant can get access to them. So that's what they're trying to show. Some of the other images here, I was trying to show that you have microbes that are interacting with the soil and the cation. So microbes, many microbes in the soil have this ability to release minerals off the soil. They can do that, whereas roots have a tough time with that. So the microbes' presence, especially fungi, they have all kinds of really interesting enzymes that can help them release and break down organic matter, release the particles off the soil. So there's really two huge ideas here. Cation exchange is one way for the minerals to get dissolved into the water, but also the action of the microbes in the soil system. So with the soil, because you have a lot of respiration going on, whoops, let me go back to this. Because you have a lot of respiration going on, the roots respiring, microbes respiring, this reaction is fairly common in the soil system. And if you remember back to basic chemistry, the pH is dependent on the concentration of the amount of protons in a system. And so because of that, if you measure most soil systems, not all, but most soil systems that have been measured, they tend to be slightly acidic because there's this fairly good dose of protons in the system. And so I'd like you to memorize, please, this pH range 5.5 to 6.5 is the range that is described as what most soil systems are found to have. And so here in this figure, you're looking at a bunch of different minerals. And then wherever you see the drawing nice and plump and fat wide, that means they're more available in that pH range. If it's very narrowed down, that means they're very unavailable in that pH range. So manganese has very low availability to the plant at a pH of 7.5, but on the other hand, down by 5.5 looks pretty good. And so if you look at 5.5 to 6.5 and kind of run your eye up, you can see that most minerals are quite available in that range. So
I think the 5.5 is most optimal.
Yeah. So again, back to basic chemistry, the pH is equal to the negative log of the hydrogen ion or the proton concentration. Is that nightmare?
Yeah.
So the minerals are more available in these pH ranges because there's enough free hydrogen ions that can do that cation exchange. And so you get nice release of the cations off the minerals. And so they're more soluble. That's the fancy way to say that. It's basically what that's saying is you got some nice cation exchange going on. The minerals get bumped off into the soil water and then they get dissolved in the water. Now the plant has easy access to those. Fungi also like slightly acidic pHs. So 5.5 to 6.5, fungi love that. So mycorrhizal fungi are very happy under those conditions. So that's great too. The downside is that both beneficial fungi and pathogenic fungi like slightly acidic pH ranges. So you do have to keep that in mind that pathogenic fungi could be also quite happy in the typical pH range. So when we grow fungi in the lab, we're always working with agar plates that are around 5.5, 5. I've even made some that are down to pH 3. There was a recipe I tried when I was a grad student, orange juice agar. That was fun to make. Take frozen orange juice and make a concoction, and the fungi loved it. So, and the pH, that one dropped it down to about pH 3. And some of the fungi I was working with, they like that. So, so here, thinking about the beneficial fungi, happy under those pH ranges, that means that they can optimize their growth. They have nice access to minerals as well that they share with the plant, as well as, of course, their finer hyphae. They're getting into narrow toxic crevices. And, this is a really nice image showing the difference. It's the fine root here. And you can see how much fatter it is than the hyphae. So even the finer roots of a plant are much bigger than the fungal hyphae. And so you have to imagine, the hyphae growing through the soil system and around all these soil particles and able to access the minerals that are on the soil particles. And so this is trying to show kind of this idea of, they're focusing here on bacteria, which I find interesting. Do you see fungi there? Am I missing it? It just says bacteria, right?
Yeah.
Okay, so there should be fungi in here. So you take this picture with a grain of salt. So there should be a lot of fungi in this system too. And what they're trying to show here is that you have the release of the cations, so they're trying to show that's happening with the presence of the microbes and doing their things. They're trying to show that the microbes can release all kinds of interesting chemicals to accomplish that. Some, they need some of that for their own nutrition, but they also will release quite a bit into the soil water, so the plant can take it up. And so the presence of the microbes in the soil system is really necessary for optimal plant growth. And it took a long time for people to realize that. And one of the things that's been happening in more conventional farming systems is a lot of the herbicides and pesticides that are applied in a continual, basis year after year. Sometimes the soil system can get devoid of microbes because of that, because it can be toxic to the microbes and they die off from it. And so then you end up with a soil system that's devoid of optimum levels and populations and ratios of the correct microbes. And then you don't have any of this going on, or very little. And so basically the plant starts to starve of the correct nutrients. And it's not just the ability of the microbes to allow the release of key minerals for the plant. They're also trying to show here that the microbes presence is helping the plant in other ways, like producing beneficial plant hormones that the plant can take, and that's a good thing. It can help the plant fight off pathogens by different behavior patterns, like producing antibiotics. They can fix nitrogen for the plant. And so the presence of the microbes are very necessary, and more now that's being realized that this is not something we should ignore. If you want to have nice, vigorous, healthy plants, that, especially in the ag world, where once you harvest that plant, you want a plant that's really rich in minerals for us to eat. We don't want to eat a plant that's so weak, so to speak, in nutrition that is going to harm our nutrition as well. So that's one of the interests I have, is looking at, ways to use microbes to boost their nutritional profile so that they can have a more dense, per gram, a more dense nutritional profile on harvest. And I think microbes are key to that, helping the plant thrive instead of just survive. This is just looking at the idea of mineralizing. The fungal presence can mineralize the phosphorus. So phosphorus, do I have that in? So phosphorus is listed on this table. So nitrogen and phosphorus, they are not cationic in form typically when they're in the soil system. They're in a negative charge. And so they can't stick to soil particles, right? They're just gonna be in the water, which is okay, because then they can be immediately taken up. But as the water percolates down beyond the roots, there goes the phosphorus, there goes the nitrogen. And you've probably heard of that huge word, eutrophication. That's what we start to see if you over fertilize a field, for example, and you way overdo it with nitrogen. It won't stick to the soil particles. It just keeps percolating with the water down to the water table, eventually to rivers and streams and the ocean. And we get too much nutrition in the water systems, and it causes eutrophication. So that's not good. And so the other idea with phosphorus, that you can get your soil tested and they'll say, oh yeah, you have phosphorus, it's all good. The other thing with phosphorus, though, that's tough, is that it complexes with other minerals in such a way that it precipitates out. And then the plants have really limited access to phosphorus. So even though you take your soil and send it off and like, oh yeah, you have phosphorus, they should always test for which version of phosphorus do I have? Do I have the type that's in the water system, or is it the type that's complexed? And the only way to break it down would be microbes, essentially. And so they usually give that profile. They'll tell you the soluble versus insoluble. And that's really, more than nitrogen, really. Plants have probably the hardest time on average with getting access to phosphorus, and then even nitrogen. So this is something that is, you can supply phosphorus to the soil if necessary. But here they were looking at, okay, what else can be going on to help plants get access to phosphorus? So here they were looking at the VAM fungi, the endofungi, and their ability to help the plant get access to phosphorus. But there's a twist to the tail here where they are saying that bacteria were also participating in this. And so, the fungi are growing out into the soil system with all their hyphae extended out. And when they were looking at what was going on, the presence of these phosphate solubilizing bacteria were there along with the fungal hyphae. And so they were doing these experiments and they were showing that the bacteria were using the fungal hyphae as like a highway system. And so the hyphae had enough liquid over their surface, over the hyphal surface, that bacteria were just kind of skating along the surface. And when the hyphae would reach a patch of phosphorus, what they were doing is in that like film of liquid that the bacteria were skating in, there was release of nutrients to foster the bacteria to keep going, I'm going to keep you healthy and fed, and let's get to that patch and start degrading it so we can get it to the plant. I mean, I'm being very loose on how I'm talking about explaining it here, but that's basically what they were finding. The bacteria could not be transferred without an energy source. So what that means is the fungi were essentially creating some food for the bacteria to keep going, keep swimming, keep skating along and get to the phosphorus. And so really a neat study. This one was published in 2021. And so I think this is one of the first studies, at least that I saw in the published literature, that they were showing this partnership between the fungi and the bacteria that both were able to solubilize the phosphorus for the plant. So let me show you this. Video. It's really cool. So yeah, once again, they're showing how you get these partnerships amongst the microbes that then partner with the plant's needs. Let me see if I can access this. Hang on. Probably won't show up in the, but here, let me. So bacteria on the fungal highway, Pseudomonas putida, moving along the hyphae of an AM fungus, VAM fungus. This is real time. Isn't that cool?
Yeah.
I just love that idea. Yeah. So this partnership, the fungal hyphae is secreting nutrients for the bacteria to keep going. Yeah. No, this is real time. It's a real time video. This is on potato dextrose agar. So through soil, it'd be slower, but here they have no obstructions. So they're zipping. They're probably like, wow, this is awesome. I don't have to dodge soil particles. So yeah, pretty amazing. So yeah, phosphorus is a difficult thing where a lot of microbes, plants to get adequate access to. So the microbes are helping out. Yay for microbes. So that, not to them. Yay, microbes. So let me go back so I can. So if you can imagine that kind of dynamic going on in the soil, and then all the other millions of microbes that are in the soil, all the mycorrhizal fungi in the soil. So now they're starting to really get some nice published studies looking at, there's been so much to the ecology of forest ecosystems in what's happening above ground, but now below ground dynamics are coming into play and telling a much more accurate and full story about what's happening in forested ecosystems. Here they're trying to show, a range of different plants, and then you have these mycorrhizal fungi growing in the soil system, and every once in a while they'll send up their fruity bodies to go through sexual reproduction. But by far all the mycelia, the biomass is below ground. And you can see they're trying to show that there's connections here of the root systems via the mycorrhizal fungi, as well as, I don't know if they're showing root grafts, but that can also happen where grafting of roots can occur. But they definitely can show that there's mycorrhizal connections amongst the different plants in a forested ecosystem, and that there is quite a bit of flow of chemicals and nutrients and information. You may have heard the term the underground highway, the fungal underground highway. And so this is what they're trying to describe and understand better of, how is this happening? What role do the fungi play? Do they have some way of controlling the flow of nutrients and sugars and minerals? Are some plants favored over others? What kind of communication is going on between the plant and the fungus? There's more and more evidence popping up that the fungi has more control over the flow amongst the plants than what was first hypothesized. But the story is definitely not finished. Like, doing research, you can always ask another question, right? And so the research continues, but they're starting to realize, at least some of the data coming in, that the fungi do have some control over who gets what nutrients and the dispersal and ratio of that in some of the forested ecosystems that they've been researching. So, yeah, so very interesting ideas. These are some of the papers that are published. So Suzanne Simard is one of the major scientists that started this work. She's out in the west edge of Canada in British Columbia, Vancouver. And she uses titles that are fairly provocative. Mycorrhizal networks facilitate tree communication, learning, and memory. And so, of course, that wrote, a lot of eyebrows got raised with that kind of title, which I always like it when scientists can push a little and get people thinking. That set up a lot of dialogue, not all of it happy dialogue. There is some pushback on that, but in my advanced botany class, which is, by the way, it's getting offered in the fall, we actually do talk about this idea of plant communication, learning, and memory. And so, and then this, I thought I like this title, Socialism in Soil, the Importance of Mycorrhizal Fungal Networks. And so very interesting work in this area, looking at what's going on below ground. Whenever I'm on Dr. Robertus's grad student committees, I'm always like pushing them to learn about, hey, you're describing a really nice interaction above ground, but don't forget what's going on below ground and how important that can be. So lots of cool work going on in this. Okay. So sadly, I'm going to shift off of fungi. It's time to move on and get into the next topic, which is, I'm going to get into lichen next. So we're not totally leaving fungi because they're part of that. And then after the lichen content, I am going to talk a little bit about algae at some point, but I want to make sure you get some bryophyte stuff before you do bryophyte stuff in lab. So I might have to shift things just a little to facilitate that. But lichens, let's think about lichens a little bit. So yes, lichens are awesome. And so here we're looking at this relationship between a fungus that provides physical protection, perhaps some chemical protection. Fungi are great at accessing and releasing nutrients off of rocks, minerals off of rocks and soil particles. They can release growth factors that the algae and the cyanobacteria like. And so the algae and CB, cyanobacteria, they can be part of this dynamic as well. So it could be a fungus and algae, a fungus and a cyanobacteria, all three can be involved. And then sometimes there's a different type of fungus, the yeast form of a fungus can also be found. And who knows what else they're going to discover when it comes to lichens. Lichens, there's still a lot of mystery to what's going on in the lichen relationship. And so the yeast is a fungus. It's just a unicellular and they don't grow with a hyphal growth form. It's just a unicellular form. And so this left picture, you're looking at all the blue, green, that's fungal hyphae. And then the algal, this lichen has algae. So all the red little dots that you see interspersed among the hyphae. So you can see how the algae are protected and sheltered and given a nice little home. All of this, by the way, up top, that's not algae. That is the fungal spores in specialized sacs called asci that are, so that's fungal spores. So kind of ignore that for this idea, all right? And then here on the right, you see with the naked eye, what we would see, and you can see that there's some nice difference in forms or morphology of the lichen. Here was a published blurb from, where is this? Purdue, where they were the ones to publish on, hey, we found some yeast hanging out with these lichens, and that's helping to explain why we see some weird differences going on. It's either the presence or absence of these yeasts that were showing some confusing results to some of their studies. And then they, like, oh, there's yeast in there, that's why. And so this finding could explain why many genetically similar lichens present wildly different physical features and why scientists have been unable to synthesize lichens in the lab, even when combining the species that partner successfully in nature. So there's a lot we don't know about lichens. Like you can't just put these organisms together in the lab and say, go to it, form a lichen. They just don't. And so clearly we don't understand them very well. And no one has ever sat in nature and watched a lichen develop. It's just you'd be there and old and gray and keel over probably before you witness that. So it's just not something that occurs in the lab or in the field that we've been able to witness. But I don't know, maybe someday they'll be able to say, yay, we synthesized some yeast in the lab. So anyways, they're very excited about this, and I like this quote from the paper: this discovery overturns our longstanding assumptions about the best studied symbiotic relationship on the planet. These yeasts comprise a whole lineage that no one knew existed, and yet they are in a variety of lichens on every continent as a third symbiotic partner. This is an excellent example of how things can be hidden right under our eyes and why it is crucial that we keep studying the microbial world. Thank you. Yes. So true. So that's a quote taken from the professor. So here is a nice picture, by the way, of, isn't this the wolf lichen or something like that? Ophioria volupina. Can't remember if that's the nickname for it. Does anyone know?
That should be fox.
Fox. Okay, I was close, but not quite. Okay, so I would like you to recognize these three genera of algae: Trabuxia, Pseudotrabuxia, Trentifolia. So that would probably be a multiple choice question if I ask it on the exam. And then Nostoc in the cyanobacterial realm. So those four genera, please know those because they are the ones that are forming this relationship. About 90% of the lichens that have been studied to date have one of those four genera. Okay, different species, but those genera. All right, so they're pretty important. The fungus, on the other hand, is kind of all over the map. They're usually ascomycetes. So far, about 98% of what's been studied fall into that. But there's some debate that maybe basidiomycetes could be forming this relationship. I wouldn't be surprised. but there's still so much mystery. Over 13,000 species have been named. They name them after the fungus. It's kind of hard. It's like, how do you name these things? So they name it after the fungus. But basically, if you can grow all these organisms separately, no problem in the lab. Like all of these are easy to grow. It's just getting them to form the lichen relationship. We haven't been able to observe that happening in real time. Yes.
So like constantly changing.
Oh, I know. It can drive you nuts.
I swear. 3 years ago, there was a different scientific name for a lichen I was studying, and I like just re-looked it up this year, and it was like, oh, it's actually this now, and I was like.
Yes, so a lot of mystery, and yeah, it's just, there's a lot to it still that we have to study. So here, this figure is trying to show this word, Soredia. I would like you to know that. Soredia. So what is that? What it is fungal hyphae that when you study it under the microscope, it wraps around either the algae or the cyanobacteria. So the algae or cyanobacteria are unicellular, and then the fungal hyphae kind of wrap around that. And this is how they disperse in nature. So if you can imagine like a water droplet or something, or a hoof, a deer hoof stepping on this, the Soredia break free and they're all in together. So you have the fungal hyphae, you have the algae or cyanobacteria that are all together and get dispersed. So that's how they kind of disperse themselves off in the natural setting in their habitat. Okay, we'll stop there and we'll continue on with the wonderful lichen.